Biology Part 103. The concentration of ACH acting on the postsynaptic membrane does not remain high for long because the synaptic cleft contains powerful cholinesterase enzymes which rapidly activate ACH, allowing the normal resting potential across the postsynaptic membrane to be restored and clearing the synapses so that the next impulse can be transmitted. There is a rapid return of ACH in the inactivated form from the synaptic cleft back to the presynaptic ending, where it is reactivated and can be reused. The effect of ACH release at the synaptic is to cause the depolarization of the postsynaptic membrane, making impulse production in the postsynaptic neuron more likely. The synapses where ACH is the transmitter and therefore described as excitatory and give rise to the excitatory postsynaptic potentials. Not all synapses are excitatory, inhibitory synapses producing inhibitory postsynaptic potentials, or IPSPs transmitters. Most neurons receive synaptic endings which are a mixture of excitatory and inhibitory types. Identifying the chemical transmitter at a particular synapse can be extremely difficult. A range of substances in addition to ACH are thought to be the transmitters. Some of these substances and their presumed effects are listed in the table. Many features of the synaptic action are still not clearly understood. For example, the details of mechanism whereby the arrival of the nerve impulse triggers transmitter release are still a mystery. And we'll move on and the role of the synapses in learning and memory, also a mystery. Well, the process is going on and science is trying to learn the things that nature has provided us. Nevertheless, the basic reason for having chemically operated synapses instead of direct electrical contacts between the neurons is quite clear. This is simply that the chemical transmitters in minute quantities are much more effective in altering the membrane potential than direct electrical stimulation by contact between the cells would be. Thus, synaptic endings can be extremely small without becoming ineffective. This means that all individual neurons can receive synapses from many other neurons, and through it the axon terminals can pass on information to many more. The possibility for an interconnection giving different pathways is almost limitless. This is one of the main reasons for the success of the nervous system in terms of information processing and flexibility. Some of the information processing and flexibility, some of the ways in which individuals' neurons can be arranged into pathways will be discussed in the next unit. We'll deal with the overall organization of the nervous system as we proceed with it. Organization of nervous system. In the tutorial, the properties of the individual nerve cells of the neurons were discussed. Evolution of nervous system. Now we consider how neurons are linked together to form the working nervous system of different animals. The simplest nervous system consists of randomly connected neurons forming an array called nerve net. Such networks are characteristics of phylum Cnidaria and Olentrates. The invertebrate groups, which include Hydra, Obelia, sea anemones, and jellyfish. When the body surface of any of these organisms is touched, nerve impulses are transmitted in all directions, causing a contraction of the muscular epithelial cells in the body wall so that the animal moves away from the stimulus. In some coelenterate, the additional through conduction pathways have developed. These are needed to coordinate the complex movements such as the swimming of jellyfish.